thanks at first for giving me the opportunity to present here a part of my PhD project. And within this talk, I would like to show you how Haemonchuc Cyp that is expressed in the model organism C. elegans can influence the ivermectin susceptibility. So as you are all aware of, Haemonchuc contortus is a gastrointestinal parasite infecting small ruminants. And infections with this parasite are mainly treated with broad-spectrum antielementics such as macrocyclic lactones. So as pictured here on the right, the most representatives are ivermectin and moxidectin, which act by permanent activating glutamate-gated chloride channels or GABA receptors in the nematode, leading to a flaccid paralysis and finally to death. So, however, the intense use of these drugs has led, as you know, to the emergence of resistance, while discovering the specific resistance mechanisms remains an important challenge today. So what is so far known about specific ML resistance mechanism is that there's no single mechanism. So we have a polygenic nature of ML resistance mechanisms is assumed, which can be divided into target site related and non-target site related ones. However, drug metabolism is by far the most important drug defense mechanisms in all organisms, as you can see by a plenty of examples in humans. So having now a look on if drug metabolism has also an impact on ML resistance in nematodes, especially Haemonchuc, Matruskova et al. Reported in 2018 that there is a constitutive expression of a UGT transferase, so a phase 2 enzyme in resistant Haemonchuc strains. But biotransformation can include a variety of enzymes, and for that, xenobiotic metabolism is divided into two phases, so phase 1 and phase 2, which are divided because of their according catalyzed reactions. So when having now a close look into cytochrome P450 mediated metabolism, which is by far the largest enzyme family for drug metabolism, and connecting this cytochromes with macrocyclic lactones, only for the human CYP3A4 enzyme, it has been linked that this enzyme can metabolize ivermectin. However, cytochromes in C. elegans hasn't been so far associated to metabolize ivermectin nor moxidectin. And for that, what I'm interested in my PhD is if a single Haemonchuc contortus gene that is transgenically expressed in the model organs can have an impact on the susceptibility towards ivermectin or moxidectin. So as I mentioned, there is only less data available on CYPs associated to ML metabolism. And for that, we first had to identify a candidate gene. And we had a look, therefore, in a study from Choi et al., who introgressed telodalzagia triple-resistant isolate into a susceptible background and performed an RNA-seq experiment. So now having a look into the data, we identified a couple of upregulated candidate genes. But when having a close look onto this specific gene, which is an orthologous to the CYP13A family, it was 20-fold times overexpressed in this resistant telodalzagia isolate. So there is an orthologous gene also in Haemonchuc, and for sure we find one. And I had a look into the literature so far and checked whether this gene or this subfamily of CYPs has been so far associated to be xenobiotic inducible. But it has been not. A study from Lying showed as well that this CYP subfamily is also only low expressed in fourth stage larvae as well as in male and female adult worms. So now to check whether this gene can have an impact on the susceptibility or not, I assembled our candidate gene into a kind of expression plasmid under the control of a Gas1 promoter from C. elegans to ensure the intestinal expression. And what I did then was I co-injected this plasmid we generated with a GFP co-marker into the gonads of the wild-type N2 strain, and then checked whether there are GFP positive transgenes or not. So GFP was only used as a marker. It is here expressed in the pharynx and just was used to facilitate selecting positive worms. Afterwards, I conducted thrashing assays, since ivermectin and moxidectin also can have an impact or affecting the locomotion of the worms. So I incubated young adults with either ivermectin and moxidectin at different concentration for 24 hours, and then counted the body bands per minute of single individual worms. So what you can see here are now the resulting concentration response curves. So here the motility in percent is plotted against the drug concentration for either ivermectin or moxidectin on the right in nanomolar in a logarithmic scale. And when having a look onto the curves for ivermectin, we're able to see that there's a right shift when incubating the transgene line with ivermectin compared to the wild type. So this means that we also have a shift in the resulting EC50 values. So in comparison to the N2 with the EC50 value of 1.6 nanomolar, the transgene is showing EC50 value of about 7 nanomolar, which means that there's a fold change of about 4. When now having a look onto the data for moxidectin, we're not able to see any shift in the curves. So no difference between the transgene and the wild type control, leading finally also to similar EC50 values. So what we can say by this graph is that this Haemonchuc from a highly resistant Haemonchus isolate, White River in this case, and expressed in the model organism C. elegans, can modulate the ivermectin susceptibility, but it does not for moxidectin, which is highly interesting. And to somehow explain this phenomenon that can be observed, I had a close look into the structures. So what you can see here are the superimposed structures of ivermectin in black and moxidectin in blue. In a binding site, so binding in a subunit of a glutamate-gated chloride chains, just to show you the differences in interactions. So what you can see here is that both macrocyclic lactones share the same core structure. However, ivermectin is possessing a big disaccharide group, leading to also somehow slightly different interactions in this glutamate-gated chloride channel. However, moxidectin does not possess this disaccharide group, but instead a metoxime group and a different alkyl side chain. So what I assume now is that there is somehow a specific substrate specificity of this Haemonchuc Cyp. And to gain further insights, I'm currently conducting thrashing assays with ivermectin aglycone, a derivative of ivermectin, which is missing this disaccharide group, to analyze whether this group can have an impact on the susceptibility or not. So in summary, we're able to show for the first time a direct influence of a Haemonchuc Cyp on macrocyclic lactone susceptibility, so specifically for ivermectin, and confirm thereby the so far reported differences between ivermectin and moxidectin in terms of pharmacokinetics. And to further analyze the ML substrate specificity of this specific Haemonchuc Cyp, I'm testing for the macrocyclic lactones. So besides ivermectin aglycone, I'm also testing doramectin, selamectin, and eprinomectin. In addition, we're also comparing the genomic sequence of this specific Cyp between different susceptible but also resistant isolates to identify polymorphisms potentially associated with ML resistance. So, and that's it. And I'm thankful for the attention, and I'm happy to take your questions as well as comments and hints for my further work. And I would like to thank Dr. Jürgen Krücken, he's my supervisor, and also DFG for funding.
Thank you very much, Natalie. So I see no questions from the Mentimeter, but my comment is that your work is very interesting and very important because we need genetic markers for macrocyclic lactones. Well, there is a question. Do you know if there are any nucleotide amino acid difference between the WR and the ICE strains in Haemonchuc contortus, or just expression differences?
In respect or in regard to this genome testing, okay, so this is what I would like to do next. So I sequenced the Cyp sequence from genomic DNA so far, and I was able to see, so I sequenced it for ISE, for McMaster isolate we selected at our institute, so a highly resistant isolate and also White River. And what I can say so far is that there are differences, so for the genomic sequence, but yeah, I know that we need to perform also additional, for example, qPCR studies between different isolates to say whether there is a difference in constitutive expression, or if there are really single nucleotide polymorphisms that can explain this effect.